My name is Caroline Wagner. I'm a pretty new assistant professor in the bioengineering department, and I work on studying how biofluids interact with pathogens in the context of understanding how diseases spread in populations. So mucus is this amazing material that we all have inside us. It coats all non-keratinized wet epithelial surfaces. So, traditionally we associate it with being sick and maybe you blow it out of your nose, but it also coats your intestine. There's a tear film of a similar polymer base that covers your eyes. The female reproductive tract is coated by mucus, so it's really acting as a protective barrier everywhere in the body. And we think that one of its main roles is to act like a very important component of the innate immune system in its ability to trap any pathogens that might get in to the surface of interest. Mucus is the gel, and then the main structural component is called mucin. So that's the glycoprotein. It's a protein backbone with a lot of sugar chains arranged radially outwards like a bottle brush or a pipe cleaner. And they swell in the aqueous solution and they cross-link to form a very viscoelastic material. The interesting thing about mucus, though, is it's this very important gel, but ultimately it's not super clear how exactly the network forms in the first place. So, when we work with some classic polymers, we can easily say how many cross-linking sites are on this material. What exactly is the mechanism for cross-linking? Is it a physical interaction? Is it a chemical interaction? With mucin, it's a bit of a black box still. We think all these interactions are relevant to a certain degree, but exactly how the network forms is still a bit of a mystery. And so that's something we really have to elucidate in order to understand how a small pathogen that could potentially be obstructed by either size-based mechanisms or biochemical mechanisms could transport through this material. So mechanical readouts are very important, again, because there's ways to connect the mechanical readout that you get to the actual structure of the network and how it forms. But now what I'm also interested in is using some more chemistry-based readouts in order to say, okay, under different conditions, so for instance, pH is a big one for mucus. The body knows that, and the eye is at a very different pH than the stomach, and the mucus behavior there, the mechanical properties and the function are very different. So what exactly about pH is causing the material to change. We have some ideas, and there's been some work on that area and some theory developed. But if we can then use a biochemical assay where we can say, all right, which bonds are different, or is the structure different, or are certain types of interactions more dominant under certain pH conditions, all of these things can help us piece together what's happening to the network. And I think now understanding the important role that mucus is also playing in terms of transmission between people, now the question is, how can I incorporate these biophysical principles into models for disease spread? Compartmental models, which operate at the population level, really think about just flows of people. So that's really, more of a macro scale picture. There is the concept of within host models where you model specific pathogen populations or cell populations, but those don't typically incorporate the real biophysical environment that these processes take place in. But we know that mucus is doing something to the pathogen. So there's evidence, for instance, that some of the sugar chains on mucins can act as sort of decoys to virus particles, since they can be similar to the ones that cells display that a virus would typically, bind to enter a cell. So we know that for some pathogens, mucus can basically just, the mucins themselves can almost trap the virus because it made a mistake and thought it found a cell receptor, and instead it found a mucin, and that sort of eliminates it from the game. It can't infect a cell anymore, because eventually what your body does is continuously clear mucus, and so whatever is trapped in the sticky mess you just swallow, and no pathogens can infect cells anymore. So those sort of processes are not taken into account, but those definitely are playing an important role in the initial stages of infection, the kinetics of those processes, and ultimately a lot of disease-based models rely on these kinetic processes, to model how does viral load evolve over time, how's that related to how much virus one person transmits, and which impacts whether or not they can infect others. So my goal is to understand better, how pathogens transport through mucus, and then incorporate that within host models for disease progression that I can connect with the population level through different sort of principles for cross-scale dynamics.
So your expertise is?
Rheology would be the classic answer, so both macro and passive single particle tracking techniques. I think I also bring to the table this expertise in the math modeling side, both in terms of analyzing rheological measurements, but also the broader modeling work that we've done. So I'd be excited to contribute to anything related to sort of those mechanical readouts, but I'm also eager to learn things related more on the biochemistry side and hopefully a bit more on the microscopy side as well.